Good afternoon, meeting on the Mesa. Thank you for hanging in there. I'm Adrienne Farid, the COO and Head of Early Development at Century Therapeutics. I'm going to change gears a little bit from the last few talks. Century is a company developing iPSC-derived NK and T-cell therapeutics in hematology and oncology. Here are my forward-looking statements. The kind of basics of Century are we're building and developing next-generation allogeneic platforms for iPSC-derived NK and gamma-delta iPSC-derived T-cell candidates. We have quite an experienced team in research and development, immuno-oncology, iPSC biology, protein biochemistry, manufacturing, and so forth. The progression that we have, I think, has been validated and exemplified by our first product, IND enabled last year in third quarter, and we're actually in our first phase 1 clinical study, so we're clinical stage at this time. We're well capitalized with a cash runway into 2026. So just a few of the pillars of the technology platform. We start actually with induced pluripotent stem cells. We have a collection of clinical grade cell lines. We have deep expertise in gene editing. This is a fundamental of our work. We're using CRISPR-Mad7 enzyme to do homology-directed repair, and I'll go into a little bit more about the precise or the gene edits that we're actually making that are core to our platform. We have a strong group in protein engineering, designing CARs, signaling and binding and so forth. And then in the iPSC differentiation, I think all of you working in that field, we have a strong group developing processes and product, and a strong analytical and characterization group. These are both absolutely critical. We actually have a wholly owned manufacturing facility, which is providing us benefits now in that we have control over our kind of clinical development timing. We're learning about critical quality attributes and critical process attributes, and will enable us to scale up and be efficient and reduce cost, which is one of the key pieces that we would like to address. We have, for our first product, we've worked with Fuji Cellular Dynamics to manufacture the first product, so they've been a strong partner to us. So just to give you a little bit illustration of the platform, we start with an iPSC bank. We do sequential gene editing steps. This actually avoids forming a lot of different variants as opposed to doing a multiplex gene editing at once. And because it's an iPSC, it continues to replicate. We isolate a single cell clone of master cell bank correctly edited cell, and that actually is the master cell bank for the life of the product. And then subsequently, we do manufacturing to differentiate into NK or T cells. So the final product in our first product is NK, but we can go in either direction. We have been focusing on what we think is a shortcoming or where there's opportunity to improve the existing allogeneic therapies. And so one of the fundamentals of all our products is we're engineering them to resist immune rejection. And so what we've shown here is, in particular with CAR NK cells, actually, they've had fairly good responses in hematology area, but the allogeneic products have had limited durability. So we think that there's a real opportunity if the cells can evade the immune system, to be able to dose more than once, and perhaps in a finite multiple dosing cycle, to actually get into durable responses. And the cells need to persist to put pressure on the tumor in that context. So we're engineering in immune evasion edits, and again, I'll go into that. So our intention is to be able to dose almost like the blue curve, where each dose is sustaining immune pressure on the tumor, as opposed to getting rejected, which more of the red curve red curve pattern. So we call this set of edits or allo-evasion edits. We're knocking out beta-2 microglobulin to resist binding by recognition and killing by CD8 T cells, knocking out C2TA to resist killing by CD4 T cells, or knocking out HLA-1, 2. And then we actually, in our first product, have put in a decoy receptor, HLA-E, which will prevent NK cell killing of our cells. Just to give you some insight, we also have follow-on allo-evasion modules. For example, what's shown here is HLA-G, another decoy receptor in addition to HLA-E, and we do a kind of a preclinical proof of concept evaluation before this would go into a product. And this shows that actually, for the most part, HLA-E is preventing NK-mediated lysis of our cells, but E plus G might end up being an improvement that we want to bring into future products. So we know the lymphoma space is crowded. We're highly impressed and amazed by the progress that's been made by the autologous products in lymphoma, the CAR T cells, but we believe that the off-the-shelf and allogeneic will end up being the wave of the future, so we really wanted to keep focusing in this area, and as mentioned, kind of change the treatment paradigm to see if multiple doses can actually get at the durable response if the cells are persisting. So our first product, Century 101, has the allo-evasion edits as mentioned on the left-hand side, HLA-1, 2 knockout, HLA-E. It's a CD19 CAR targeted cell. We have a safety switch, which is a short EGFR repeat, and we have engineered an IL-15 to enable cell persistence. And the cell does have 6 gene edits, and we had a very useful dialogue with FDA in the pre-IND interactions, and our IND, as mentioned, was cleared. Just a little bit of the preclinical data. We challenged the cells with lymphoma cell lines on the left-hand side. There's in vitro study showing serial killing versus NALM-6 and Ray lymphoma cells, and the Century 101 cells continue to kill over 7 cycles, depending on the cell line. On the right-hand side, there's a tumor xenograft lymphoma study, a mouse study that shows tumor cell control, tumor growth control with both fresh and cryopreserved cells. So just some of the preclinical data. We're in the midst of running a phase 1 clinical study. We're looking at Century 101 in patients with relapse refractory CD19 positive B-cell lymphoma. The study is called Ellipse 1. The primary goal are safety, PK, preliminary efficacy, and so forth. We're in as a standard phase 1 study. We're in the dose escalation portion of the study. We're escalating first in a single monthly dose from 100 million to 300 million to 1 billion cells. By the end of the year, we expect to be dosing at the 1 billion level. Then we move to, in addition, we're evaluating a second schedule, Schedule B, which is 3 doses per month on day 1, 8, and 15. There's another important design aspect of the study. If patients have stable disease or better, they actually can get additional cycles of treatment, so additional monthly cycles. So we actually have the ability to look at multiple dosing, both in a monthly cycle regimen or through the 3 times per month regimen. With regard to lymphodepletion, we're interested in being able to evaluate whether lymphodepletion is actually really required if these cells are immune evasive. So for the first 2 cycles that a patient would get in this trial, they will get lymphodepletion, and subsequent cycles they will not. So we really have the ability in the study to look at whether the cells are persisting and are putting tumor pressure on in a few different multiple dosing regimens with and without lymphodepletion. We have a thorough translational program associated with the study. So one of the things we thought was important, because CAR-NK products have been found to traffic very quickly out of the bloodstream, we're not only looking at PK in blood, but we're looking at cell-free DNA coming from our cells, and that will indicate trafficking or persistence within the body, not necessarily in the bloodstream. And for those of you not as familiar with cell-free DNA, which it's used for MRD analysis often, DNA is a very short half-life in the body. So if the cell DNA can be found in the body, in the analysis, it indicates that the cells have trafficked to a place other than the bloodstream and tissue distribution. We're also looking at a kind of a comprehensive immunogenicity characterization, looking at both cellular and humoral immunity, looking at anti-drug antibodies, CAR-neutralizing antibodies, ADCC and CDC. We also believe that the PK, whole body PK almost, will give us some indication whether the alloevasion is working and the cells are persisting and can be routinely persist. We're looking at tumor biopsies and looking at tumor microenvironment, recruitment of immune cells to the TME, and of course other PD markers like minimum residual disease, B and T cell kinetics, and safety biomarkers like patient cytokine levels. So we're looking forward to release the early data set of clinical and translational data later this year on this trial. We're very excited about that. In addition, in our pipeline, which I will just go over very, very briefly, we have a follow-on gamma delta T cell product with dual targeted CD19, CD22 that we're interested in looking at more broadly in B-cell malignancies. Century 107, a Nectin-4 targeted IT product for a series of solid tumors. And we have an ongoing, very productive collaboration with Bristol Myers Squibb, where we have 2 products directed, one at AML and one at multiple myeloma. And they may be either NK or T. So to conclude, we're very excited about being able to work in the space of iPSC-derived therapies. We're excited to be able to learn extensively in this current trial that we're running and actually apply the learnings to subsequent products and move the field forward together with all the rest of you. Thank you.